Yeah, sure. So, hi, my name is HCT. I'm a junior at Notre Dame San Jose High School.
And hi, my name is Ojasvi. I'm a junior at Evergreen Valley High School.
And our research focuses on the in-silico characterization of potential serum response factor inhibitors in colorectal cancer. And our research advisor is Mr. Kunha. Here's a general outline of our presentation today, and Ojasvi will start with our background.
So colorectal cancer is a cancer which starts in the colon or the rectum. It's the second most common cancer leading to death and the third most common cancer in the US. And this is why, just due to the need, as shown by these statistics, this is why we chose colorectal cancer as a cancer to focus on for our in-silico analysis. And on the right is the picture of our HCT116 cells, which is a colorectal cancer cell line. So SRF, Serum Response Factor, is our target, and it's an important transcription factor which binds to a regulatory DNA element known as the CARG-G box. This binding is shown on the left figure, and it's involved in two crucial signaling pathways involved in growth, which is shown in the right figure. The classic pathway, which involves stimulation with growth factors, and the row-dependent pathway, which involves changes in actin, and these two pathways cause the activation of SRF, which results in transcriptional activation of growth-related genes, which impacts various cell survival and survival versus cell death, and also cytoskeletal elements like actin, which have to do with cell migration. Sorry. Okay. So based on the fact that it impacts survival and also kind of cell motility, we chose to kind of hone in on how SRF can impact cancer. So as shown in this figure, SRF has been shown to downregulate E-cadherin, which leads to the epithelial-mesenchymal transition, which leads to metastasis. So the epithelial-mesenchymal transition is basically when so in this, I guess, the normal state, the cells have an epithelial kind of morphology. So that basically means that they're normal. And when this transition occurs, the cells kind of change their phenotype. They become more migratory, more stem cell-like, and mesenchymal cells are stem cell-like, which allows them to metastasize, leading to poor outcome. So in general, E-cadherin prevents this from happening by allowing from contact inhibition. So that's basically when the cells reach a certain mass, they stop dividing. So in cancer, SRF has been shown to downregulate E-cadherin, leading to a loss of contact inhibition, allowing for the epithelial-mesenchymal transition to occur, leading to metastasis and poor outcomes, and this has been seen in gastric cancer and colorectal cancer. Based on these factors, we decided to look into SRF as a potential target for colorectal cancer. So far, there were several SRF inhibitors developed. Shown on this slide are our two positive controls. So CCG1423 is an SRF inhibitor associated with a better response to enzalutamide, which is a prostate cancer drug. And the drug on the right, CCG22740, is an SRF inhibitor used to treat fibrosis. So based on these known inhibitors, we will investigate other potential SRF inhibitors found through virtual screening of the Campbell database and docking analysis through various, along with determining kind of the properties of the drug through other computational methods like molecular dynamics simulations and pharmacologicals or toxicity analysis, which Akshati will go into more detail about.
Right, so I'll be talking about our methodology. And this is a general pipeline for our methodology. We've just completed our molecular docking, and we're working towards the other steps like molecular simulations and drug cell line prediction. So the first step in our methodology is data collection. We've used the Campbell dataset, which has over 2 million compounds. And Campbell has a feature called the similarity search. So for any given compound, you can search for the similar compounds at a specific similarity threshold, like 40%, 50%, etc. We've used the 40% similarity threshold. And using the two positive controls that Ojasvi mentioned, we use this similarity search to get a list of 84 compounds that are 40% or more, at least 40% similar to the positive controls. And the Campbell similarity search uses a Tanimoto similarity algorithm. And the Tanimoto similarity algorithm basically scores compounds against each other for their chemical and structural similarity. And out of the 84 compounds, some of the compounds had missing values or null values for some of their properties. And in our next step, which is clustering, these compounds would have thrown errors. So we had to remove those compounds from our list. So we ended up with a list of 76 compounds, which we used for our next steps. And we also did run the Tanimoto similarity algorithm on our end with the Campbell dataset to just confirm this list of 76 before we moved on. So the two clustering techniques that we used were k-means and HDV scan. So k-means takes in a parameter n, which determines the number of clusters that the data will be grouped into. And to determine this optimal value for n, we use something called the elbow technique. So shown on the slide is our elbow plot for our data set. Basically, the elbow plot shows the amount of variance for each value of n. So as we can see here at n equals 1, or if there's just one cluster, there's a high amount of variance in the plot, in the clustering. But as we get larger numbers like n equals 2, 3, 4, and so on, the amount of variance decreases greatly and starts to level off. So there's not much difference in the variance at 2 clusters and 3 clusters and etc. So we determined that n equals 2 was our optimal value for the k-means plot, and our graph will be shown later in the results section. We also used HDV scan to cluster our data, and HDV scan is unique in that it doesn't put noisy data into the clusters. So basically, any outliers in the data set are not put into the clusters, which so basically HDV scan has a less likely chance that clusters might be skewed. And both k-means and HDV scan are clustering techniques, but our data has many dimensions. So to be able to visualize the cluster plots, we need to reduce the number of dimensions to 2. So we used UMAP, which is a dimensionality reduction technique, to be able to visualize these graphs. And so from our k-means and HDV scan clusters, we picked out 10 compounds, which Ojasvi will go more into in our results. And with those 10 compounds, we performed the molecular docking. We used Autodock Vina for molecular docking. Basically, Autodock Vina visualizes how a compound, how well a compound would bind to our target, SRF. And it gives the binding affinities for each compound. And Autodock Vina requires that each compound, ligands and the receptor are in the PDB-QT format. So we use OpenBabel to convert all the files to that format. And shown here is SRF with its active site highlighted in blue, and we visualize this with a P2 rank and PyMol. So now we'll be going into some of our preliminary results.
So this is our clustering data with the HPD scan clustering on the left and the k-means clustering on the right. So overall, 2 clusters were found. The drugs, which as Akshati said, were narrowed down to a list of 10, which were common for between both clusters, were used for further docking analysis. And the drugs in both of these are found in the brown cluster.
And the figure to the left shows our best candidate visualized as binding to SRF. And the table shows 4 of our candidates, which are highlighted in purple. with their binding affinities, as well as our 2 positive controls highlighted in green and DMSO highlighted in yellow, and DMSO was our negative control. And as we can see, 2 of our candidates did have better binding affinities than both of our positive controls. So those were our top candidates.
So this figure shows the protein ligand interactions for our top 2 compounds shown on the previous slide, and our 2 positive controls in the binding pocket. And this figure was generated using Autodock tools. So you could see the amino acids which the drugs bind to in their specific binding positions. And this figure will be going into more detail in the discussion.
So in addition to clustering and molecular docking, we've also been working on de novo drug design using a software called Autogrow 4. So Autogrow 4 kind of works in 2 parts. So first, it takes in a list of compounds and it docks them to a target with Autodock Vina or some kind of molecular docking algorithm. And then it works to improve those compounds to have better binding affinities to the target. So far with Autogrow, we've gotten a couple of compounds that have slightly better binding affinities than our candidates from the clustering, from the Chembl dataset. So shown here is one of our best candidates, which has a binding affinity of negative 8.2. So it's not much better than the best candidate from before, which was negative 7.9. But we're still working with Autogrow to hopefully make better compounds.
And I guess now going on to our discussion. So overall, of the drugs downloaded from the Chembl dataset, Chembl 4073009 and Chembl 4088815 have the highest binding affinities. And based on the binding analysis done before, the common binding residues between the top 2 drugs were arginine 164, lysine 163, and glycine 167. And both arginine and lysine are positive amino acid, and so it makes sense that these amino acids would be present in the active site of a DNA binding enzyme, as DNA is negatively charged, which further validates the drugs. And overall, this data could potentially indicate that these residues are necessary for drug-ligand interactions, although a lot of further investigation would be needed. Furthermore, the new drug from Autogrow has an even greater binding affinity of negative 8.2 kilojoules per mole. It has a similar structure to the top 2 drugs with several carbon rings, and its properties do need to be investigated further using our future steps.
Yeah, so some of our future steps. So currently we've started working with deep CDR, which predicts the drug sensitivity on cell lines. And we'd also like to use the liver MLDL tool, which predicts liver toxicity, as well as PRED-HERG, which predicts cardiac toxicity. And we're also getting started with GROMAX, which is a molecular dynamics simulation tool. Basically predicts how a compound will interact with its environment when it is actually put into a cell. And eventually, we'd also like to synthesize our top candidates and test them in the wet lab on HCT116 cell lines, and possibly looking into altering the residues of the SRF active site that help it bind to DNA, and hopefully by altering those residues, inhibiting its binding to DNA. We'd first like to thank our mentor, Mr. Kunha, for helping us with this project, as well as the new group for giving us the Autodock Vina tutorial. And finally, we'd like to thank ASDRP and the Olive Children Foundation for giving us this opportunity to conduct this research.
These are references. And thank you for listening. You can scan here to view our references further. And any questions?
Great presentation. Thank you.
I guess we have a question. How will you scan through the database for potential candidates?
So are you referring to like the Kemble database? Okay, so for the Kemble database, first we used Kemble's similarity search because Kemble has over 2 million compounds. So to kind of narrow that down, we use a similarity search, which also uses Tanimoto similarity algorithm. And so that gave us about 84 compounds to work with at a 40% similarity threshold. And so with those 84 compounds, we were able to do clustering. And after clustering, we narrowed it down to 10 compounds, which we use for molecular docking.
Did that answer your question or? Okay, thank you. Any other questions? So what will you do after knowing which compound is most effective / best at treating the HCT116 cells? So I guess what we would do once we reach that point is ideally we would start expanding. First of all, we would look into, I guess, we would compare like the toxicity of the HCT116 cells with like normal epithelial cell lines. And if that shows good results, obviously we'd also have to compare it with known treatments. And then based on that, we could start again expanding to organoids and to like mice models, hypothetically, if we reach that stage. And then hopefully we'll be able to gain insights on whether this is actually feasible as a treatment for humans. But it takes a lot of work to get from this stage. And then first we'd have to confirm it all computationally, and then we'd have to actually synthesize it. And then we'd have to do wet lab experiments comparing RNA expression, the actual, I believe, MTT assays, etc. Western blots, again, to see whether this actually succeeds in changing like gene expression and to kind of characterize how this impacts the cells. And then we'd have to move on. So it's a long pipeline. Did that answer your question? Thank you. Are there any other questions?
Thank you both for presenting. And yeah, I guess if you do have any other questions, you can always reach out to us even after the presentation, and we can still talk about it. But yeah, I think that's all we have at the moment. So thank you all for listening, and yeah, have a great Tuesday. So a great week. So yeah, if yeah, thank you everyone.
Thank you.